Every single cell has a nucleus, and every single nucleus has all the DNA a particular organism needs. Just to repeat that, every cell and nucleus has all the same DNA as every other cell in an organism. The difference is that not every gene is turned on. Cells of different tissues perform different functions, so they demonstrate different genes. These genes provide the instructions for making different proteins that your body uses to perform all kinds of functions. Not every cell end up making every protein, but each one starts out with the same potential to do so. This is why, when a parent cell divides, all of the DNA in its nucleus is replicated. Human cells have 46 chromosomes, 23 pairs of chromosomes, and they need to be replicated each time the cell divides so that the daughter cells get an exact copy of the parent cell. For grade 10 science, we say that a cell carries out all its usual activity during interphase. It's more complicated than that, but not for our intents and purposes. During interphase, all the organelles in a parent cell are duplicated, just like the DNA. Mitosis is the splitting of the nucleus, which leads to the division of the cell as a whole. Normally, all the DNA in the nucleus, or chromatin, can be found in long strands all tangled up. Fun fact, each strand of chromatin is about 5 centimeters long. However, when the cell begins its division stage, the nuclear membrane of the cell disappears, and the chromatin has condensed into 46 chromosomes and can be seen under a light microscope. This first phase of mitosis is called prophase. During this time, two organelles called centrosomes head toward opposite ends of the cells, and thread-like tubules called spindle fibers begin to form. These spindle fibers eventually stretch out from the centrosome and attach themselves to the centromeres that are attached to the middle of each centrosome. Now, look at the diagram. The two centrosomes at each end of the cell, the spindle fibers are attached to all the centromeres, and now the chromosomes are starting to line up across the middle of the cell. This is the longest phase of mitosis, called metaphase. It is important to note that each centromere around the middle of each chromosome has two spindle fibers attached to it. The shortest phase of mitosis, anaphase, begins when the proteins that attach the two chromatids together at the centromere break apart. The spindle fibers, which are stretched like elastic bands, retract and pull each chromatid, which is half of a chromosome, towards its respective end of the cell. Once the chromatids separate, each one becomes a chromosome in its own right, and the cell now has twice as many chromosomes as usual. So now the spindle fibers have pulled the chromosomes to opposite ends and begin to disappear. Because we have entered the final phase of mitosis, telophase, membranes begin to reform around each group of chromosomes again. A nucleolus appears, and the chromosomes uncoil and become harder to see. It should be noted that the cells begin to stretch and pinch in at the center. This is what marks the beginning of the end. The cell membrane continues to pinch in around the center until the parent cell splits into two daughter cells. The daughter cells get half the cytoplasm and half the organelles from the parent cell, so now it has a full set of chromosomes, a full set of organelles, and all the cytoplasm it's going to need. It should be noted that cytokinesis is not technically part of mitosis, but is added in the presentation because it offers some closure to this lesson on mitosis as the main part of cell division.